Hey everyone, welcome to Gray's Gyan. Today we're diving into intermediate filaments, the unsung heroes of the cytoskeleton. By the end of this video, you'll know exactly how they're built, what types exist, and why they matter clinically. Let's get into it. So what exactly are intermediate filaments? The name itself gives you the first clue. They are about 10 nanometers thick, intermediate between the thin 7 nanometers actin filaments and the thick 25 nanometers microtubules. Now here's what makes them fundamentally different from the other two cytoskeletal components. While actin and microtubules are assembled from globular proteins that bind and hydrolyze nucleotides like ADP and GTP, intermediate filament proteins are filamentous monomers, and they completely lack enzymatic activity. No nucleotide binding. No hydrolysis. Remember this for your exams, it's a classic MCQ. If proteins have no enzymatic activity, unlike G-actin, ADPase, and tubulin, GTPase. Alright, now the most important part. How are intermediate filaments actually built? It happens in 3 beautiful steps. Step 1. Dimer formation. Each If protein subunit has a characteristic structure, a central alpha helical rod domain of about 310 amino acids. That's the core, flanked on both sides by non-alpha helical head and tail domains of variable size. 2 of these subunits wrap their central rod domains around each other like a coiled coil, forming a parallel dimer. Both proteins point in the same direction. The head and tail domains are the variable parts. They determine how F proteins interact with other cellular structures. Step 2. Tetramer formation. Now 2 of these dimers come together, but this time they align in an antiparallel, half-staggered fashion. One dimer points one way, the other points the opposite way. This creates a tetramer. This antiparallel arrangement is critical. It's why intermediate filaments are anoenpolar. There is no plus end or minus end, unlike actin and microtubules. Step 3. ULF final filament. 8 of these tetramers then associate laterally, side by side, to form a unit-length filament, or ULF, which is 16 nanometers thick. These ULFs then anneal end-to-end, joining together longitudinally. As more ULFs join, the filament undergoes internal compaction, narrowing down to the final mature intermediate filament, 10 nanometers thick. Herman et al., 2007, a landmark paper that defined this 3-step assembly model using electron microscopy. Excellent reference for deeper reading. Now that we know how they're built, let's talk about what properties this gives them. Look at the illustration from Gray's Anatomy here. You can see the tetramer, the unit-length filament, and the mature filament. Property 1. Intermediate filaments have incredible tensile strength. That tight association of dimers, tetramers and ULFs makes them highly resistant to stretching, compression, twisting and bending forces. Think of them as the structural steel of the cell. Property 2. They are anoenpolar. Because of that antiparallel tetramer we just discussed, there are no preferential ends for growth. Property 3. NO nucleotide binding, unlike G-actin which binds ADP, and tubulin which binds GTP. And property 4. ULFs join end-to-end with each other, in both directions. This is unlike actin filaments and microtubules where the plus end always grows faster than the minus end. Where are intermediate filaments found, and what do they actually do? First, structural strength. They are found in huge numbers in cells that face mechanical stress, nerve axons, muscle cells, epithelial cells. Look at the fluorescence image here from Gray's Anatomy. Those green networks in the keratinocytes are keratin intermediate filaments, and in the nerve section you can see the neurofilaments. Second, they form extensive cytoplasmic networks, from cage-like arrangements around the nucleus all the way to the cell surface. They're literally everywhere inside the cell. And third, this is the clinically crucial one, different intermediate filament proteins are expressed in different tissue types. This makes them powerful molecular markers in histopathology. Pathologists use them every day to identify the origin of tumors and the degree of cellular differentiation. Intermediate filament proteins are classified into 5 types based on their primary structure and tissue expression. Let's go through all 5. Type 1 and Type 2, keratins. Found in epithelia. always form heteropolymers, always an equal combination of a type 1 acidic keratin and a type 2 basic or neutral keratin. There are about 20 types of each. Type 3, vimentin, desmin, GFAP, porphyrin. These form homopolymers. They can assemble on their own. Vimentin is in connective tissue, desmin in muscle, GFAP in glial cells of the brain, and porphyrin in peripheral nerves. Type 4, neurofilaments, nestin, syncytin, alpha-internexin. Neurofilaments are the dominant cytoskeletal protein in neurons, especially in axons. They are heteropolymers of NFL, NFM, and NFH, low, medium, and high molecular weight subunits. NFL is always present, combined with either NFM or NFH. Type 5, nuclear lamins. Lamins A, B1, B2, and lamin C, which is actually a splice variant of lamin A. These line the inner surface of the nuclear envelope in all nucleated cells. They form anastomosing networks, not linear bundles like the other types. A great clinical tip: carcinomas express keratins. Sarcomas express vimentin. Brain tumors express GFAP. This is used daily in diagnostic pathology. Let's zoom in on keratins because they have important genetic disease associations. Within the epidermis, keratin expression actually changes as keratinocytes mature. In the basal layer, cells express keratins 5 and 14. As they move up to the spinous or prickle cell layer, they switch to keratins 1 and 10. This matters because genetic defects in different keratins cause disease in different layers. Disease 1, epidermolysis bullosa simplex. Caused by mutations in keratins 5 or 14. Remember, these are the basal layer keratins. So basal cells become fragile. They lyse after minor mechanical trauma, causing blistering of the skin. Reference: Porter and Lane, 2023. Disease 2, epidermolytic hyperkeratosis. Caused by mutations in keratins 1 or 10, the spinous layer keratins. The spinous layer cells lyse, causing intraepidermal blistering. The skin becomes thickened and scaly. Rule of thumb: basal layer keratin defect equals epidermolysis bullosa simplex. Spinous layer keratin defect equals epidermolytic hyperkeratosis. This is a classic exam question. Type 3 proteins. Vimentin is expressed early in development in mesenchyme-derived connective tissue cells and some ectodermal cells. It is also used as a cancer marker. Vimentin-positive tumors are typically sarcomas. Desmin is the IF protein of muscle, both skeletal and cardiac. Desmin mutations cause desminopathies, including cardiomyopathies. GFAP, glial fibrillary acidic protein, is specific to glial cells and is a critical marker for diagnosing gliomas and glioblastoma in the brain. Porphyrin is found in peripheral nerve axons. Type 4, neurofilaments. In axons, neurofilaments are so abundant that they are literally the dominant protein. NFL plus either NFM or NFH, always present in combination. When neurofilaments accumulate abnormally, they form neurofibrillary tangles, a hallmark pathological feature of Alzheimer's disease and many other neuropathological conditions. Nestin is a type 4 IF protein found specifically in neuroectodermal stem cells. It's a stem cell marker. Type 5, nuclear lamins. Lamins are unique. They are in the nucleus, not the cytoplasm. They line the inner nuclear envelope, providing a mechanical framework for the nucleus itself and acting as attachment sites for chromatin-organizing proteins. Mutations in lamins cause a group of diseases called laminopathies. These include Hutchinson-Gilford progeria syndrome, the premature aging disease, as well as Emery-Dreifuss muscular dystrophy and dilated cardiomyopathy. Reference: Burke and Stewart, 2013. Before we wrap up, let's quickly acknowledge the key researchers whose work forms the foundation of everything we've discussed. Harold Herrmann and colleagues at the German Cancer Research Center published a landmark 2007 paper describing the three-step IF assembly model, dimer, tetramer, ULF, using electron microscopy. Essential reading if you want to go deeper. Penn et al. In 2012 gave us a comprehensive review of keratins in normal and diseased epithelia, the molecular basis for all those blistering diseases we discussed. Porter and Lane in 2023 updated our understanding of how genetic keratin mutations affect epithelial mechanical stability. And Burke and Stewart in 2013 reviewed the nuclear lamins, critical for understanding laminopathies. I'll link relevant resources in the description below. Here's a comparison table to lock in the differences between the three cytoskeletal components. This is pure exam gold. Diameter, actin 7 nanometers, intermediate filaments 10 nanometers, microtubules 25 nanometers. Monomer, actin uses globular G-actin, IFs use filamentous proteins, microtubules use globular alpha-beta tubulin dimers. Nucleotide binding, actin binds ADP, IFs bind nothing, microtubules bind GTP. Polarity, actin and microtubules are polar with faster plus ends and slower minus ends. IFs are non-polar. Assembly, actin and microtubules grow from the plus end. IFs grow by ULFs joining end-to-end in both directions. Enzymatic activity, actin has ADPase activity, microtubules have GTPase activity, IFs have none. If you can reproduce this table from memory, you're ready for any exam question on cytoskeleton. Now let's see how all of this plays out clinically. Here's the key concept. Because if proteins are tissue-specific, they serve as molecular fingerprints, telling us exactly where a cell came from. In cancer diagnosis, this is invaluable. A pathologist sees a metastatic tumor in the lymph node. Where did it come from? If the tumor cells are keratin positive, it's a carcinoma, from epithelial origin. If they're vimentin positive, sarcoma, from mesenchymal origin. If GFAP positive, it's from the brain, a glial tumor. Genetically, mutations in IF proteins cause serious diseases. We covered the skin blistering diseases from keratin mutations. We mentioned neurofibrillary tangles and Alzheimer's from abnormal neurofilament accumulation. And laminopathies from lamin mutations, including the devastating Hutchinson-Gilford progeria syndrome, where children age rapidly and die in their teens from cardiovascular disease. GFAP is routinely used in neuropathology to identify gliomas. It's one of the first immunohistochemical stains a neuropathologist will order. Let's quickly summarize. Intermediate filaments, 10 nanometers thick, assembled in 3 steps, dimer, tetramer, ULF, into nonpolar filaments with no nucleotide binding and incredible mechanical strength. 5 protein types, each tissue-specific, making them critical diagnostic markers. And genetic defects cause real diseases you'll see in clinical practice. If this video helped you, hit like and subscribe, it genuinely helps the channel grow. Drop your questions in the comments, and I'll answer them.